Hey, Nick, how are you?
Hi.
Should get everybody here in a second. There we go. Hey, Melinda. Hey, Marcella. Got everybody. 1, 2, 3, 4, 5, 6. Hi, guys. Looks like 5, 2, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7. We're missing one. Who are we missing? We're missing. It's okay. We got Nick. We got Sarah, Marcella, Sophie, Katie, Sydney. Just trying to figure out who we're missing here. I think Jenna. Which Jenna's with us? Burr or Kiss?
Yes.
Okay, got it. So we're missing Jenna Burr, and that's it. All right. Cool, guys. All right. We'll go ahead, and I don't want to waste your time because I hear that you've been Zooming a lot, you guys, you honors people. So let's go ahead and start real quickly with a prayer, especially for today, Election Day. Father, Son, Holy Spirit. Amen. Lord, be with us today as we enter into a virtual time together. Thank you for the opportunity to be together, even if it is virtually. Please protect our minds and our hearts today. Fill us with peace and joy, and always help us to focus on your Son. Amen. Father, Son, Holy Spirit. Amen. Okay. So I haven't Zoomed with you guys before, but let me find out why the screen isn't showing. You guys can see the screen, can't you? Thumbs up. Let me get my view here. Boop. There we go. Cool. All right. So I think I can see all you guys. Feel free to keep your video on or off. It doesn't matter. And what I wanted to point out first, first let me ask you if you got any questions so far. You guys okay? Any questions?
No.
Okay, good. Let me also ask you before we get started about the AP Classroom. I know one person for sure couldn't log on to AP Classroom, and I don't want to do anything really on there until you guys all get registered and logged on. So is anybody else, there was one person who contacted me about that. Anybody else having troubles logging on to that? Nope.
No.
Okay. Shoot me an email if you have trouble logging on to that. If we end up going past Wednesday with a virtual learning, I might dig into a little bit more, but we'll see. Right now, we've got enough to keep us busy. So I just wanted to check in with you. I've also added what we're going to talk about today, cell communication. I've added that to your Google folder. So let me show you that. If you go to your cells, there we go. Cells and division folder. It is right here. Cell communication. So right there is what we're going to talk about today. If you guys want to log on to that. Totally fine. All right. Oh, here may be Jenna. Oh, there we go. Cool. All right. So I won't talk at you a whole lot today. I'm going to try and keep it to like 25 minutes or so of talking at you, but this is being recorded. So I'm also going to upload it to Google Classroom so you can kind of get a little bit more interaction if you want to go back. We're not going to get through this whole thing today because this is the basis of a lot of what we're going to do in the future. And you're going to be working on Thursday and Friday, whether you're in class or whether you're at home. You're going to be working on something to help you learn this material, the cell communication stuff. There's a lot of new terminology. So feel free to stop me at any time if I'm like blowing through things too quickly, or if you just got questions, I'll stop and ask you questions as we're moving along. Give me a thumbs down or let me know if you can't hear me well. Okay, I'm gonna assume that you guys can hear me. I can hear you pretty well too. So let's get started with cell communication. Again, this is on, Jenna, you just joined us, I think. Cell communication, this is on your Google Classroom main page stream, and it is in the cells folder. So cell communication is in there. It's so nice to see you guys. Yay, I'm so excited. Okay, let's get started because I know you got a lot to do today. So first things first, I'm going to probably be looking both ways to see if you've got any questions and then also to let you know what's going on here. So we've got two vocab words right off the bat. One is a ligand and one is a receptor protein. So the ligand is the molecule that is going to come in and attach to the receptor on the cell. So in our example of what's going on with this coronavirus, in this case, the ligand would be the virus itself, that little coronavirus shaped thing. Boom, 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 boom. And here is our lovely lung cell that it likes to hang out with. And it has on it a receptor protein on the outside. And the ligand, this guy right here, is going to attach to the receptor protein on our lung cells. The virus doesn't actually enter our cells, but it injects its DNA into our cells and communicates and takes over the cell that way. So a ligand is the signaling molecule. It can be anything, it can be a hormone, it can be whatever, any kind of different chemical. And then the receptor protein is what it binds to. So here we've got a receptor protein right here, and here's my ligand. And a lot of times we have to use these receptor proteins and ligands because we said that the membrane in here is, it likes water or it doesn't like water. Do you guys remember the inside of the membrane right here, the tails, do they like water or not like water? Good, shake your head, they don't like water. So the reason that we have a lot of these proteins embedded is because this layer is hydrophobic and a lot of our ligands are hydrophilic. And so they attach and then they're going to send their message inside. Sometimes they can attach and something happens here at the protein. You're going to do this G protein pathway kind of a deal. We'll walk through that a little bit later. And so the message, the ligand never goes inside, but the message does. And you use like a secondary messenger system. But other times, if I'm a hydrophobic ligand, I can actually go through. So a lot of our steroids are hydrophobic. They can go right through the membrane and then they attach to a receptor inside the cell. So here's outside the cell, here's inside the cell. Outside of the cell, I'm hydrophobic. I can't go in. I got to send my message through the protein. I'm hydrophobic down here. I can go right through, but I'm still going to bind to something inside the cell. And I'm still going to have some kind of cell response based on my ligand, whether it goes directly in or whether it binds to the cell on the outside. 4 different ways that cells can communicate with each other. One is that they're touching each other, and then they can send messages back and forth. The second one is these two are a little bit long distance signaling. I'll talk to you more about those. And then the synapse is nerves. So this is when they're right next to each other. We're going to use other things, other ligands, and it's a little bit more long distance. And then we've got a synapse where nerves are either communicating with other nerves or nerves can be communicating with the cell directly, as in the case of like a nerve telling a muscle cell to go ahead and contract. So 4 different ways that cells can communicate. And we're going to go into detail about each one of those. Direct, paracrine, endocrine, and synaptic. Again, let me know if I'm going too fast for you guys. All right, direct contact. Oops, sorry. If you didn't get all 4 of those written down, I'm going to go ahead and go over them individually. So let's dig into direct contact. You can see right here that the plasma membranes of these cells are touching each other. And then you've got these proteins right here that are allowing molecules to pass from one cell to the other. And sometimes this happens a lot with like heart cells communicate this way. So cardiac cells are touching each other. They're sending signals from cell to cell. They don't have to go very far. They're passing the message all along, almost kind of like the telephone game, because they're touching each other and they can interact like that. So direct contact is when they're touching each other. Example of that would be a cardiac cell, set of cardiac cells. Let me know if I'm going too fast. And again, this is also on Google Classroom. Paracrine is when I'm not touching, but I can release my little ligands right here. These green guys are ligands, and I'm going to release them into, say, a blood vessel. So that blood vessel is going to take that message and go long distance, short distance. So sometimes this is a long distance, sometimes this is a short distance one. But an example of this would be like your thyroid gland, and it's going to release thyroid hormone, and that's going to go into the bloodstream of the blood vessels around the thyroid gland, and it's going to go to different parts of the body. Your thyroid is a big regulatory gland for all kinds of metabolic activities. And so when the thyroid releases something, usually the effects are kind of widespread. So this is paracrine signaling. I'm making something like a hormone or a steroid or something like that. I'm releasing it to nearby areas like a blood system, circulatory system, and it's going and it's affecting other cells down the line. Next one is endocrine, and this is even more specific with just hormones, where my hormone is usually released like in the thyroid gland and other glands. They've got the blood vessels right next to it, and it's going to go to target cells around the body. So let me give you another one for paracrine, although endocrine is a type of paracrine signaling. This might be, let's see, I'm going to say, let's go with in the kidneys when I am making or releasing urine. That's a great example of the paracrine signaling. And I'm releasing things into the body through the kidneys. So endocrine specifically is glands and hormones. And synaptic is my last one. So this is when you either have a nerve that is ending with another nerve and it's going from one nerve to another, or my nerve is ending at a cell like a muscle cell. And the nerve cell is transmitting an electrical impulse, turning it into a chemical impulse with neurotransmitters. And those neurotransmitters are being received either by a cell or by, you can also have from nerve to nerve. So the neurotransmitters are things like acetylcholine, epinephrine, things like that. And they are chemicals that tell the cell what to do. So for example, epinephrine is released and it tells the heart to speed up. So this might be a nerve cell going and taking epinephrine and going to the heart cell, and then that's going to tell the heart cell to speed up. But it's involving a nerve cell. So synapse is only specific to nerve cells. It doesn't relate to anything else because the synapse is between the end of one nerve cell and either the beginning of another nerve cell, if I'm going on down the line and just shooting my message down, or if I've got a cell at the end that I'm giving a message to. All right, so this is the part that we're going to spend a little bit of time on. You've got your ligand, and then you've got your response at the other end. And in between, you've got a whole bunch of stuff that can happen depending on what kind of ligand it is, what kind of cell it is, what kind of response you're looking for. So signal transduction, that's just everything that happens inside of the cell between the reception of the ligand and the cellular response. So there's a whole lot of things that can go on here. We'll talk about a couple of them today, but not all of them. I'm gonna make sure that I'm keeping you guys on time. And so just know that signal transduction is anything that happens in the cell between the ligand binding and the cell response. So there are a couple different things that we're going to look at, maybe just two today that we'll get through. You guys okay with that one? All right, we're going to get into some terminology. A lot of different terminology that you probably never heard of before. Maybe phosphorylation in chemistry. Not sure if you might have heard about that then, but the words seem confusing, but what they do actually isn't really that confusing. Their definition isn't too terrible. So these terms we're going to throw around actually quite a bit. So if you write them down now, that's going to help you, but we're going to definitely go over them again and again and again. So don't feel like at the end of today, oh my gosh, so many new terms, I got to know all these. We're going to spend about a week on cell communication, whether we're in school or we're distance learning. And so you're going to have plenty of time to practice using these. Not a big deal. So what is phosphorylation? It's just adding a phosphate group. We'll talk about that more in a minute, so don't necessarily worry about that one. But what is a protein kinase? We're going to talk about these a little bit later. They're enzymes that help this process of getting the signal from the ligand to the cell response, because cells obviously have no way to communicate other than by touch. So things moving around inside of the cell. This protein kinase is just an enzyme that in the presence of something else, it says, ooh, it's time to do this. We'll talk about specifically how that happens in just a second. And then phosphatase is also another enzyme. What these do is either they're going to add a phosphate group, and that's this guy right here. It's a negatively charged molecule that's used for a lot of things. We use it in ATP, we use it in GTP, which we'll talk about later. We're using it in this process of phosphorylation. And if I'm adding a phosphate group through phosphorylation, I'm going to use this guy. This enzyme is going to add my phosphate group onto the end of the molecule, which then signals something to happen. Or if I've got a phosphate group and I'm taking it off, in the instance of GTP, which we'll talk about a little bit later, if I'm taking my phosphate off, that again sends another signal that something else needs to happen in the cell. So all these adding and removing of phosphates, all that does is it tells certain other things in the cell, oh, it's time to do this, or it's time to stop doing this. So some cell signals that we have are actually stopping mechanisms, and maybe you don't want to produce any more of a certain hormone, or you don't want to produce any more of a regulatory protein. And so you can do that. You can regulate it by either adding a phosphate group to block it, or taking the phosphate group to send a message to stop it. So phosphates are used quite a bit in the cell communication business, and they're just negatively charged particles. If you remember, I still have your photosynthesis video pulled up so I can show the freshmen at some point. But when you talked about ATP in photosynthesis, ladies, ATP is just adenosine with 3 phosphates at the end. And when you lose those phosphate groups, that gives off energy. So phosphates are used a lot in cell communication and other processes of the body. So phosphorylation is we're working with phosphate groups. I'm adding a phosphate. If I'm going to add it, I'm going to use this particular enzyme, which we'll talk about later, protein kinase, and the cascade of what that happens. And then phosphatase is the other one, and it removes a phosphate group from some molecule or another. And we'll talk about specifically what that means, but I wanted you to be familiar with these two before we dive into, okay, what's going on inside the cell between the ligand and getting my cellular response. So we're starting off general. We're going to get more specific here in a second. This is just the shape of these, showing how ATP is having a phosphate removed to become ADP, and phosphatase is used to do that. That's all this is showing. All right, so back at the way beginning, I showed you that there was an intracellular receptor, and that's when my ligand can go through the membrane. And then my cell surface receptor, that's the one that's actually embedded in the membrane, and then the receptors on the outside of the cell, so the ligand never actually enters. So two receptors, they both do the same thing. They both attach to a ligand or touch a ligand. They're not going to bind, they're just going to touch, and that causes something to happen. So one is inside the cell, one is on the cell surface. They both have to receive the ligand, and the ligand is the message. I'm going to say ligand about 1 million times. You're going to get really tired of it, but then that word's going to stick out in your brain for you, because the ligand starts everything off, and the ligand can be pretty much anything. They're usually, I can't even think of another time that they're not proteins, but they're proteins that come in, and because of their shape, I'm shaped like this, my receptor is shaped like this, and so I touch and something happens. So here's my ligand of a certain shape, here's my receptor protein, so they're both proteins, and because they've got that specific shape, they're going to touch, and then something's going to happen. All right, we're at the membrane now. So we're at the membrane level. I've got 3 kinds of receptors if I'm within the membrane. So we're not inside the cell yet, but we're hanging out here at the cell membrane. I've got this one called channel linked. Ions like hydrogen ions or chlorine ions can go from one side to another. I've got a receptor that's an actual enzyme itself. And then this is the one right here that we're going to spend a lot of time on, because I think out of all of these, it's the most complex. So if you understand this and the way this goes, this messaging system, this G protein pathway, you can explain any kind of cell communication to anybody. So we're going to actually focus on, and you're going to choose a different G protein pathway to explain. I'll talk to you more about that in a little bit. But G protein pathway just means you got several different proteins actually that are being used to get a message from outside of the cell to inside of the cell. And we'll talk about what GTP is here in just a second. So you've got 3 kinds of receptors. These are in the membrane, embedded in the membrane, and they're sticking on the outside. Channel, ions are going through. Enzyme is activated when a ligand touches it, and G protein pathway. And that's the one that we're going to spend a lot of time on. So if you just want to write the names of those down, we'll go into detail about maybe 2 of them. And then tomorrow we'll do the G protein pathway, because that's the one that you guys are going to focus on, either in groups or in partners. Let me ask you this and give you a starting point while people are still writing. So how many of you would be comfortable working on like a paper-based project via FaceTime or something like that with someone who's not with you? Like if I put you in partners, or you get to choose your partners, how comfortable would you be with working with like a paper-based, so you can show me and move around and you're working together on it? Would that be too complicated for you? You guys okay with that? Okay. Because I found that over the years, the best way to have you guys learn about cell communication is to create a G protein pathway. And that would be, you would choose a specific ligand and cell response, and then you'd create paper cutouts and you'd move them around and work through the process. And it takes several days to kind of figure out how to do that. But if you can do FaceTime and either both of you working with paper or one of you working on the paper with the cutouts, and then you're both explaining it, then that'll work. And if we get into it and it doesn't work, or if we're back face to face on Thursday, not quite sure that's going to happen, guys. We got a lot of people out. So I'm just giving you a heads up. It's not official, so don't say anything to anybody, but we haven't heard a word yet officially on whether we're going to be back Thursday or not, but chances are pretty slim, because I looked at the number of people who are having to quarantine right now, and it's a lot. So just giving you a heads up. You probably knew that anyway, but I'm just preparing you for that in case that happens. All right. So here's a picture of an example of each one of these. So here are my ions, and they could be, like I said, here's a good example, hydrogen ions, where we're creating ATP, and we're going to add some phosphates, and the ions have to move from one side to another. Here's my ligand. Notice that this is closed, this channel protein. Here's my membrane. This is closed until I get my ligand to touch it. Boom. And that says, ooh, open the door and let me in. All right. And I'm going to let some ions in. Here's my second one. This is, that's the G protein pathway. That's enzyme 1, right? Yes. So this second one is I'm going to create a response. So all I'm going to do is bind as my ligand. And because I've activated a protein, so that's this one, my enzyme receptor is right here. So I've activated my enzyme, and it's going to do something within the cell. So my ligand binds and my enzyme is, ooh, now my enzyme's active. And when my enzyme's active, other proteins within the cell are going to do something. They're going to move somewhere. They're going to make a protein themselves. Lots of different things can happen with this enzyme activated. And then C, this is C, both of them. This is the G protein pathway. So it looks a little complicated. You're like, holy sheep dip, this looks really complicated. You will actually be able to explain this in about a week. No problem, right? I promise. So your ligand comes here. Here's my G protein receptor. And we're going to go into detail about this. So don't worry, I'm just giving you an overview. So in this case, could be adrenaline is actually an example of one. So adrenaline is my ligand, and it touches the receptor protein on a particular cell, maybe a liver cell, and my liver cell is going to break glycogen down into glucose. My cell response is I want to flood the body with glucose. So here's my adrenaline. It touches this G protein. Because this G protein has a certain shape, when this touches it, it changes shape. I've got an alpha, a beta, and a gamma subunit that are on the inside of the cell. So outside of the cell, inside of the cell. And when, here we go. So my alpha subunit, my beta and gamma stay right here attached to or right next to this G protein. My alpha subunit detaches and it moves. And it doesn't use energy from ATP. It uses energy from GTP. Same concept. So what is that molecule that I'm using right here that's going to create energy? I just told you about it about 4 minutes ago. So what is that molecule that's coming off to create that energy? It's negatively charged. Anybody remember? Starts with a P. Ends in phosphate. Phosphate. All right. There you go. So my phosphate group is going to, when that pops off a GTP, that gives us a little bit of energy to move this guy within the cell. And then a lot of different things can happen. So we can have a protein kinase cascade. We can have all kinds of things that happen, but the beginning of it is going to be the same each time. A ligand will come in, it binds to this changes shape. These two guys release this moves over to adenylyl cyclase is usually this guy right here, and that's going to allow something else to happen. So because this one's a little bit more complicated, that's the one that we focus on, because with all the moving parts, if you can figure that out, you're in like Flint. Let's see how we're doing here. Give you about another 5 or 6 minutes or so. So I'm inside my cell now. That was all outside of the cell. If I am a steroid, I can go right on through the membrane. And why can I go through the membrane if I'm a steroid? Remember what our membrane looks like. Why can I go directly through if I'm a steroid? I don't need it outside.
Hydrophobic.
Yes, so steroids, exactly. Bingo, thank you for playing. So steroids are hydrophobic. And because they are, they can go right on through there. So they're lipid soluble. And usually what hormones do is this guy right here. What does that mean by gene expression? If I say I'm going to express a gene, Think back to bio days. We haven't covered it yet. What does it mean when I express a gene? You guys remember from bio? Everybody's saying no, they don't remember. What is a gene? Not blue jeans. What's a gene?
Like a DNA sequence?
Exactly. So in your DNA, the only reason you have DNA is it's a code for making proteins. So when we want a gene to be expressed, we want a certain protein to be made. That's all that means. It sounds fancy because we've put so much hype on genetics and gene technology and DNA technology, but it just means that that stretch of DNA that codes for a certain protein, it's turned on so that protein's made. That's it. That's all DNA does. It tells proteins to be made, or it stops the production of proteins. It's a shutdown mechanism. So DNA either makes the protein, tells the body to make the protein, or tells the body to stop making the protein. So I'm going to show you a picture here in a second. If I'm a steroid and I'm going to go through into the cell, I have to have a place on me where I'm going to bind. I also have to have a place on me that's going to go into the nucleus of the cell and bind to that DNA. And I have to say, turn on, meaning DNA, it's time to make this protein. So I got to be able to do two things. I have to be able to get inside the cell, which I can do easily. I just diffuse through because I'm hydrophobic. But once I'm inside the cell as a steroid, I have to go somewhere and either get a carrier to take me into the nucleus of the cell, or I can just go in there directly. But I have to be able to bind to that DNA. If I'm a steroid and I just float around inside of the cell, there's like no point whatsoever for me being in there. I've got a job to do. So in this case, I have to go into the nucleus. I have to find that specific segment of DNA that says, make this protein, and I've got to bind to it. And then I've got to say, okay, it's time to go. So three things, three different shapes make a steroid what it is. Being able to get in there is easy, but I got to be able to find something inside the cell. I've got to be able to get inside the nucleus and find my correct spot of DNA. This is all miraculous, by the way. I think this is amazing. And then I've got to be able to say to that DNA, okay, it's time to go. We're going to turn on, which means you're going to copy that code. It's going to leave the nucleus, and I'm going to make myself a protein. So let's take a picture of that. Here are my little mushroom guys. These are hormones. They slide right on through the membrane. Inside, they've got this little complex right here. And normally, I don't want this particular protein to be made. And so this is blocked. Here's my inhibitor right here. And this little protein right here that looks kind of like a polar bear sideways. So he's on this area right here. This molecule is what I need to get inside of the nucleus. So normally this is like inactive, done. I'm not doing anything. I'm not telling the DNA to make this protein. I'm just a molecule that's waiting for this little mushroom guy to attach to me. So I'm a hormone. I go into the membrane. I touch this area right here. This is the binding site of the receptor protein inside the cell. Once I do this, my polar bear, my inhibitor comes off. My inhibitor is there all the time. When he's removed, oh, this says, oh, I got to take this hormone and I got to take it and transport it inside the nucleus. So this little guy right here has a DNA binding site attached to it. It's going to go through the holes that are in, here's my nucleus right here, my DNA is inside. It goes through now because it's been activated, my inhibitor's been removed. I go inside through the nuclear pores and I find the DNA part that matches with this, that's part of this receptor protein right here. I'm inside, and then I've got another part of me that says, oh, it's time to turn this gene on. It's time to make this particular protein. And so my cell response is to express a gene. So I've gone from my ligand, which is a particular hormone, a steroid, and my cell response is to make a protein. That's what gene expression means. So it's fairly complicated, but not nearly as complicated or as involved as what G protein pathway is. This is inhibited normally. My little mushroom guy binds. It says, oh, the inhibitor can leave now. Now I can go, my mushroom guy is going inside, attached to all this complex right here. Inside, attaches to the DNA and says it's time to make a protein. Holy smokes. Let's talk about these and then we'll wrap up because that's a lot for you guys today. So what are these guys right here? You're going to see the word kinase used a lot in the next week. So do you remember what a kinase is from like 8, 9 slides ago? What is a kinase? Anybody besides Katie? Dare to answer. Let's see if I can get to it. Talked about it way back here.
An enzyme that adds a phosphate to a protein.
There you go. Bingo, bango, bongo. Awesome. So kinase is just, it's another name, a fancy name for an enzyme. And it's also going to bind to a ligand. So now we're at the membrane receptor. Adenylyl cyclase is an example we'll use tomorrow. So I'm a receptor now. And when the ligand binds to me, so this is a separate scenario here. We've moved past our hormone. Now we're going to be in the membrane and we're going to receive our ligand with the kinase itself. And we're going to do this funky thing here. Dimerization and autophosphorylation. Oh my gosh, on a Tuesday, we are doing hard things. So this is a funky word. It really is not that big of a deal. It's going to be the movement of 2 molecules. This is not a big of a deal either. Phosphorylation is we're going to work with phosphates, and auto just means it's doing it by itself. So these words look scary, but they're really not when you see a picture of them. So go ahead and write this down, and then I'm going to show you what the picture is of this happening. And then we will wrap up, and I'll see if you've got any questions. So receptor, if I'm a tyrosine kinase, I'm going to receive my ligand. I'm going to be activated by this process right here, which I'm going to show you, dimerization and autophosphorylation, which means I'm going to add a phosphate group. And then something else is going to happen. So I'm in the membrane now. And I'm going to do these 2 processes to make something else happen inside the cell. I'll have to show you a picture of it here in just a second. All right, so our example here, write this down at the end, insulin. And if you know anybody with diabetes, you know that insulin is super important. They might need it. What do we need insulin for? Anybody have a clue? What's up with insulin? Why do we need it? Diabetics.
Doesn't
out
it
there.
help?
You know anybody?
Yeah.
What do we need insulin for? It has something to do with sugar, but do you know what?
Is it blood levels?
Yes, I think you said sugar blood levels. Insulin regulates our sugar blood levels. Insulin is the molecule that lets sugar inside our cells. So the reason why it's
Did it stop for everyone else?
Yeah.
I bet the Wi-Fi at the school just went out or
At-
something.
Crash.
Okay, I was gonna say,
Yeah. That's
sorry,
all
did you guys get back? Are you guys back?
yeah.
Okay. Sorry about that. This happened yesterday too. I kind of need to be here because I need the board, but the internet totally just crashed yesterday and booted us all off of everything. So we're all like, whatever. It's Corona. It's the election today. It's bad vibes in the air. All right. So insulin is our example here, and this will be the last thing, and then we'll wrap up and I'll let you guys go. So what does this mean? All right, this fancy word right here, dimerization, and this fancy word right here, autophosphorylation, just gonna explain them to you right now. All right, so here is insulin. In this example, my ligand, this guy right here, is an insulin molecule. So I've got two proteins that work together. They need to be paired like this. Normally, they're super close, but they're not touching each other. There's no communication. So when my insulin binds, I've got tons of insulin like flowing all over, produced by which organ? What's our insulin produced by?
Pancreas.
Bingo. Awesome. So my insulin touches these two proteins right here. Normally they're hanging out, they're chilling. All this fancy word means, dimerization means that these move together and they add some phosphates to them. Autophosphorylation, I'm adding phosphate groups. And dimerization means I move closer together. That's all those fancy words mean. So normally my proteins are hanging out. They're like in the chill phase. They're doing nothing. They haven't been activated yet. Insulin activates them and tells them, ooh, touch each other or get super close and add some phosphate groups to yourselves. That's it. That's the message. Now, because of that, here's what happens. So if this is the only thing that happens, it'd be pointless, right? So like, who cares? We got phosphate groups attached to these little receptor proteins. Who gives a rat's rip? Well, over here, because these phosphates are now on here, I've changed shape. So here my protein was normally like this rod shape. Now I've changed it to have these little phosphate groups attached to the side. Because I've done that, this little guy, the little polar bear looking guy, comes up, and it could be, depending on what it is, it's another protein, a secondary messenger, and the protein now recognizes it, picks up a phosphate and says, ooh, I need to go do something else. So in the case of, let me see if the next one, yep, here we go. So that's the basic part of it. And let me tell you about insulin now. So I've done this with insulin. Here are my specific proteins that are going to receive insulin. I've dimerized, I've autophosphorylated, and now this receptor protein right here snatches a phosphate and it goes, this is in a liver cell, it goes to where this molecule called glycogen is hanging out. Glycogen is like extra glucose that you eat that your liver stores for a hot second in case of emergency. For example, before your extra sugar, if you ate 5 Snicker bars, some of it's going to be stored as fat because nobody on the planet can burn off 5 Snicker bars. I can imagine, like maybe an ultra marathoner, maybe, right? But any extra sugar, your liver takes up and says, I'm going to hold it here in the form of glycogen for a hot second because you might get chased by a bear and you might need to like sprint all of a sudden out of nowhere. So insulin is going to go to the liver cells. And it goes all over the body and does lots of things. But in the liver cell, this is a liver cell. In the liver cell, this dimerization and autophosphorylation causes this protein to find those glycogen molecules and say, the body needs glucose, not glycogen. It needs glucose. So it tells the glycogen when it binds to it, touches it, break down and throw some glucose into the cell because we're going to need to run. So this is one pathway right here. And then this fancy terminology of dimerization, it just means I'm moving together. Autophosphorylation means I'm adding a phosphate group. When I do that, this certain shape change causes this guy to come find me, this receptor protein. He picks up a phosphate. This protein now has a job to do. It goes to find the glycogen and says to the glycogen, time to break down, we need you. Now, if you don't need the glycogen from the 5 Snicker bars that you ate, the extra sugar, it's going to be stored as fat. But the liver has this magical job of providing the body with that extra sugar. Even if you might have eaten 8 hours ago, you ate those 5 Snicker bars 8 hours ago, the glycogen from them, the extra sugar, is still in your liver. So what you ate hours ago, your body can use when it needs to, which is good, because if we only use the sugar or the energy that we ate in that moment, we wouldn't be able to do very much, right? We have to be eating all day long. So this cellular response process is actually very brilliant. And to me, you just can't get this from nowhere. This like doesn't randomly evolve. I keep saying that over and over again, but come on, people. You can't think that this is random. So these molecules are causing the response of glycogen breaking down into glucose. And that's where we're going to stop today, because that is one heck of a lot. So somebody tell me in their own words what it means, dimerization. What does it mean to dimerize?
When two molecules are moved closer together.
Yep, that's it. Sounds scary. It's not. And autophosphorylation. What am I doing there? Come on, someone besides Katie, because go ahead.
Adding a new phosphate group.
Yeah, you're just adding that little PO4 group, and that causes a change in the shape. So what could you say about the importance of the shape of proteins in cell communication? If I were to ask you to just summarize kind of everything we looked at today, how important is it that we're using proteins to do this stuff for us? And why are proteins so important? You can't mess this up. I'll help you.
It's necessary because their shape, like they need a certain shape to bind with the other things.
If I were here, I'd be throwing some candy at you right now. Awesome. Yes. So the reason why all this can happen is because proteins have different shapes, and different shapes interact with other shapes. So that's all this is. Cell communication is just proteins touching each other, touching me, touching you. That's Sweet Caroline, right? That just popped into my head. Squirrel. So it's all about shape and proteins. And you're going to dig into G protein pathway a little bit more tomorrow. I'll give you some examples of that. And then for several days, whether you're here or whether you're home, we're going to be working on you're going to choose a specific G protein pathway from ligand to cell response. And you're going to be able to explain it. And I'll explain more of that tomorrow. All right. Any questions from the peanut gallery? Are you like, holy cow, this is crazy, insane? Are you guys okay?
What was the project that you mentioned?
So what you'll do is you will either on your own, depending on your comfort level, or with a partner. If we're in school, we're going to grab some big paper and you're going to create a G protein pathway, and you're going to be using little cutouts to walk through kind of what I just did on this one. You'll be able to do that. You could choose insulin and breaking down glycogen into glucose as your ligand and cell response. But what you'll do is you'll create on paper. It doesn't really work digitally unless you're like super fancy pants and you can make these things move. But what's really getting it into your brain and helps you understand this is if you can create these little green cutouts to represent insulin, you're going to draw a membrane, but everything else is going to be a cutout so that it can move, so that you can show dimerization, you can show autophosphorylation, you can show this receptor protein moving to glycogen and glycogen breaking down into glucose, kind of like your own little animation, but using paper. And once you're able to do that, I'll give you a day or so to practice it and be able to go over it again and again and again. And if we're here in class, what I would have you do is when you're ready, you'd call me over and you and your partner, one of you would just explain the whole process, just kind of like what I did just now. Insulin is the ligand and it binds to the receptor protein, and that causes dimerization. And that process of the two of them coming together causes a phosphate group to be added on each of these proteins. This receptor protein takes a phosphate. It then goes to the glycogen synthase, which causes glycogen to be broken down into glucose, and that is your cellular response. You'll be able to do that by the end of the project. Everybody will. Everybody. So that's really what the project is. How about what are your predictions on whether we're going to be here on Thursday or not? I'd like to know what you guys have to say. What do you think?
I don't know. I don't really know anything. We don't really know anything.
Yeah,
I
we
feel
like
like you
don't
guys
know.
are.
You
They
can
haven't
go.
told us anything. We don't know anything about. Everyone says the cases are rising, but we haven't heard anything.
We don't
Okay.
like, yeah, because we don't actually know how many kids at Newville have it. Like they just like, they
That's
haven't even
true.
said anything. They just say like, oh, there's more, but like,
Yeah, not like a number.
Well,
So-
yesterday, obviously I can't share names, but yesterday when I looked on the list, because we got a list of everybody who's quarantining so that we could plan, there were 97 people quarantining, 97 students.
Yeah, well, the
Now, that
school
was
is also
yesterday
saying.
morning.
That's like the majority.
There's
of
no
the
point
schools.
in
That is the
97
football team
quarantine.
and volleyball team.
if we're taking
The football
out,
team is not quarantined like the volleyball team.
Yeah, but right now we're quarantined. We're supposed to be.
if we're taking Monday to Wednesday off, like, why don't we just take the rest of the week though?
Yeah.
I think that's
Yeah.
what's gonna come down. I can't tell you yes yet because we haven't gotten the official notification from Mr. DeCuff. So when I walked by his office yesterday, I mean, every time I walked by to like bring something down, because we gotta bring seating charts down so they can trace every new positive. I know of 1, 2, 3, at least according to what I got this weekend, about 4 freshmen are newly positive.
Yeah, that's
least
what my
that's-
sister said. She said it kind of went crazy.
Yes. And so it's kind of spread. Now, what does that mean? Does that mean that everybody's got symptoms? No, it doesn't sound like it. I got a direct email from one freshman this weekend that said, hey, just wanted to let you know, my dad's positive and I probably am too, but my test results are going to come back. I don't really have any symptoms, but I kind of have the no smell, no taste thing, but like, that's it. So was she confirmed yet? I don't know that, but from what she said on
No,
the email,
she's not.
right. So that's the kind of thing right now where we don't want to say a whole lot of things like, oh, all these people are positive. What we know right now, though, is according to that list that I saw yesterday morning, I'm sure it's grown since yesterday morning, at least 97 students, and then look to be about 9 coaches, maybe 7, 8, 9 coaches, and 1 teacher are quarantined.
Oh wow.
So that's a lot of peeps.
Yeah,
It's
not even
weird though,
working.
because like the CDC says one thing and the school says another though.
This is all driven right now by the health department. So Mr. DeCuff and Mr. Lynn have no say in who's quarantining or for how long, or even anything about the school. So if you have so many cases, I've told you guys this before, you have to shut down, which is what we've done. Well, now what that's allowing us to do is to what I think, this is my prediction of what's going on. What I think is happening is that we're getting input from the health department and they're going to tell us the new open date based on the last contact date of the last positive person who was at school. So we were all here last Tuesday, right? That's the last time any of us were exposing anybody else probably at school. So in my mind, 2 weeks from last Tuesday is this coming Tuesday. Now, I don't know if that's what we're actually going to do or if it might be a little bit longer than that. But what the health department does is they take the schools out of the picture. So it's not Mr. DeCuff and Mr. Lynn making these decisions. It's them processing the information that they're getting constantly. Because with 97 people quarantined, that's a lot of data to manage, right? So they're making decisions for not only are we open, but how are we going to handle conditioning? How are we going to handle this event? How are we going to handle, this and this down the road? So that's what they're doing right now based on what the health department, who's in charge now, is telling the school. Because once you have so many cases, it's like it's out of your hands. And the health department, at least in Michigan, I don't know other states how they're doing it, but in Michigan, the health department comes in and then it's like out of your hands. Does that
Yeah,
make sense?
the only reason I said that is because when the CDC called, they only called some of the volleyball girls, but when they called some of us, they said, well, your quarantine's done the 18th, or not the 18th, the 8th, but the school isn't letting us come back to school till the 10th. So it's just kind of annoying because it doesn't seem like anyone's on the same page.
And that's probably because from the time you got that phone call until the time they did more tracing, more information came out.
Well,
So
I
what
found
the-
out I couldn't come back to the school till the 10th before the CDC even called me.
Oh, okay. It really, when you should get a final thing from the school saying, this is what we're doing. And the reason it's confusing is because your return to school initially might have been based on an initial positive case, but then there's been an additional case and you might have had contact with this person like later on. And so now the school has to process all these recommendations by the health department and then create some kind of a global plan as to when everybody can come back. So the health department might have called and said, okay, this is your date, but then a couple more cases and they haven't just called you back yet. Because imagine, I mean, I can't even imagine right now having to call all these people that are being traced. How do they, like, what the manpower that's involved in that is insane. So I'm sure there's going to be some miscommunication here and there. But I do know that the school has to follow whatever the health department recommends and that they're the ones doing the tracing. Like the school just gives them the names of people that sat around the other people. And then they like put it in an algorithm and figure out what's going on. At least that's what I've been told. Could be slightly different, but it's kind of chaos. It's a little foobar, if you know what foobar means. But you know, it'll be fine. Eventually we'll get back. The goal is to get back, but also the goal is to kind of work together and make sure that we're part of the solution and not part of the problem. Because if you've been paying attention to the local numbers, like the hospitals are starting to get crammed again. The death rate seems to have leveled off a little bit though. So even if you're being hospitalized, it's no longer a death sentence, which in March it kind of was. If you were put on a ventilator, chances were 90% or so that you were going to die. And now it's like much better. If you are hospitalized and put on a ventilator, now with all the new treatments and stuff, it's actually a more decent chance that you're going to make it out of there if you are reasonably healthy to get, in the first place. So I don't know. I don't watch too much TV, so it's too crazy. Can any of you guys vote? Have any of you voted?
Yes, I can. Jenna.
Yeah.
I will be today.
Okay.
Yeah, I'm going today.
Cool. You're going today? Good for you guys. Yeah, I'll be interested to see how that kind of plays out with all the drama and stuff about the election. So that's great though that you're going out to vote. It might take you a while, but depending on where you have to go. But kind of exciting. Something else to focus on, I guess, besides the virus. What's going on?
Ugh.
It's a beautiful day. Go outside, hopefully at some point. It's supposed to get like in the 60s, I think, right? Did I hear that?
This
don't
week
know.
is supposed to be like the 70s.
Yeah. What?
Wow.
In November? I'll take it.
Yeah.
We need to get outside. Cool. All right. So we're going to end this today because I know you probably have other Zooms that you've got coming up and I want to be respectful of your time. But tomorrow we'll finish this up and then I'll also give you a little bit more heads up. Hopefully, I would imagine that by tonight, we're going to get something in the email about more definite what's going to happen Thursday or Friday. I just wouldn't count on coming back Thursday or Friday. So what we'll do is we'll tackle this project from home virtually and then we'll just work through it together. So it'll all be fine. It's always fine. Right? All right. You guys have any last minute questions? If not, we'll wrap up with prayer.
No, I'm good.
Nope, you're good? All right. It's great seeing your faces. Maybe we'll do, when we get a little bit more, oh, sorry, go ahead. Do you have a question?
Do we have
Okay.
a Zoom?
Well,
Do we have a Zoom tomorrow?
Yes, so Zoom tomorrow. It'll be more of this. All right, no quick quizzes or anything like that. Just we're gonna talk and go slowly. And maybe we'll do either this week or early next week. If we're still doing virtual, we'll do some fun, like, I don't know, like bring your pet to Zoom or like a scavenger hunt, or we'll do some fun stuff too. So we'll do some fun stuff. It won't be like this all the time because ain't nobody got time for that. That's a lot of Zooming. All right, let's wrap up with prayer. In the name of the Father, Son, and the Holy Spirit, amen. And if we could pray together a Hail Mary to blanket the nation in protection and intercession. Hail Mary, full of grace, the Lord is with thee. Blessed art thou among women, and blessed is the fruit of thy womb, Jesus. Holy Mary, Mother of God, pray for us sinners, now and at the hour of our death. Amen. Father, Son, Holy Spirit. All right, thanks for popping in, favorites. I'll see you guys tomorrow.
Bye.
Bye.
See ya.